Good evening, everyone, and welcome to today's wonderful evening with Dr. Murali Bharadwaj. We are now in the mania of mind maps. One of the biggest challenge for every neetpg aspirant, how to bring enormous information consolidated into a easily capsulated format. That is what mind maps will help you. You analyze, summarize, visualize, and memorize. So these are the typical steps for you to have a long-term memory for the tomorrow's neetpg exam. So in this pursuit, the whole 50,000 high-yield facts, all examiner, any number of exams, any number of years, ultimately will play with these 50,000 high-yield facts. You need to do the revision. So how is that possible to do a rapid revision, assimilating the concepts? That is through the mind maps. So we have come up with the e-books, which contain typically around 150 to 200 mind maps per subject. Each mind map revises around 8 to 10 high-yield facts. And that is the fastest way that you can become the topper. Now, what you are having here below is the link. Once you click on that link, you can go to the PDF of the microbiology snapshots, where you have a PDF with 150 mind maps, which I am discussing now. And very fast, 2 minutes, 3 minutes, you can be able to revise one mind map. And that's how within 7 to 8 hours, you can be able to finish one subject revision. Is it not what we are dreaming all the days? Yes, sir. So let's make the great beginning of the revision with the microbiology mind maps, microbiology snapshots. So, doctor, can the online students can punch? Is the voice loud and clear for all of you? Now I will fade out and I want you to have the screen fully for you to focus. So, pigment producing bacteria. This is the favorite topic of the examiner. And also the spore forming the bacteria. This mind map fully focuses and gives you a quick review. Let me pick up the pen. Which color do you want? Blue. Okay. Pigment producing bacteria. Actinomyces israeli. It produces, this is a little thicker. Let's take a little thinner one. Okay. Yellow sulfur granules. Very classical thing is yellow sulfur granules. And Actinomyces israeli are the filaments of the bacteria. Staphylococcus aureus. It also produces a pigment. The very name aureus means gold. So golden yellow pigment. Then Pseudomonas aeruginosa is another pigment producing bacteria. It produces blue-green pigment called pyocyanin, pyoverdin. Then Serratia marascens. It typically leads to what is called pseudohemoptysis. Please remember, pseudohemoptysis. It produces a red pigment is what you need to remember, doctor. So, yes. I hope my voice is loud and clear for all of you, right? And also the screen is very clear. Good. I'm happy to see that. Now, Staphylococcus epidermidis, What is the speciality? So there are certain bacteria. A lot of times the question is asked about which one among them produce the biofilm. So in all the catheter and prosthetic device infections, it is the staph epidermidis. Similarly, in the dental plaques and infective endocarditis, it is the viridans streptococci, which includes Streptococcus mutans, Streptococcus sanguinis. Then the respiratory tree is colonized, especially in the cystic fibrosis and ventilator-associated pneumonias, by the Pseudomonas aeruginosa. Then contact lens associated keratitis is also caused by the Pseudomonas aeruginosa. Similarly, the non-typable uncapsulated H. influenza is also a in vivo biofilm producing bacteria that typically lead to otitis media. So a list doctor. Now comes what are the spore forming bacteria. So some gram positive bacteria form spores, especially when the nutrients are limited. जब energy supply कम होता है, then they form the spores. And these spores, they lack any metabolic activity. They are highly resistant to heat and chemicals. And in the spore, if you take, it contains dipicolonic acid, and that is the one which is responsible for the heat resistance. How do you sterilize the spores? Only autoclaving can kill. Almost 121 degrees Celsius for 15 minutes is required. And hydrogen peroxide and iodine-based agents are also sporicidal, is what you need to remember. So ultimately, you need to know what are all the examples of the spore-forming bacteria, doctor. Spore-forming bacteria. So you should remember that anthrax, Bacillus cereus, which lead to food poisoning. This has got a short incubation period. Only 1 hour is the incubation period of Bacillus cereus. Similarly, Clostridium botulinum, which lead to botulism. Clostridium difficile, that lead to pseudomembranous colitis. Clostridium perfringens, that lead to gas gangrene. Clostridium tetani. These are all the examples of the spore-forming bacteria. Abhi aage na? What are the pigment-producing bacteria? What are the in vivo biofilm-producing bacteria? And what are the spore-forming bacteria? Whatever. Now you are the champions for the tomorrow's exam, doctor. So Pranati is saying, hello sir, not able to download, is it? You can, doctor. See, Pranati, you go to that link and it will ask you to pay 400 rupees. Once you paid, the download link will open, and from that you can be able to download the PDF. Okay? So that download link which I have given you now, so there you need to go. It has got Instamozo store. In that you can be able to download the PDF. Don't worry. Any problem is there, we will definitely help you, Pranati. You can WhatsApp me. And my number is 9502119082. You can WhatsApp me. I'm always here to help you to get the PDF. So no worries about it. So can the online students can punch? Is the voice loud and clear for all of you, doctor? Yes. Good. Now I'm once more going back to the focus on the on our mind map. So flagellum, what is it? Flagellum is a protein structure which is used for motility. All of you know. Now, there is something flagellum, other is pilus and fimbria. So pilus and fimbria have glycoprotein, unlike flagellum which has got only protein. Pilus and fimbria mediate adherence of the bacteria to the cell surface. And the sex pilus is important for conjugation is what you need to remember. Now we have come to a very important area, doctor. Few important points about the bacterial structure. So if you look at the spore, typically there is a slime layer which has polysaccharides, and the slime layer helps to adherence of the surface and also important for the biofilm formation. Now in the bacterial capsule, it protects against the phagocytosis. We have something called opsonins. If you look at the complement proteins, the terminal components of the complement proteins are opsonins. They're important for membrane attack complex they form and break the capsule. They protect against the phagocytosis. And bacterial capsule is made up of polysaccharides and rarely proteins. Then if you look at the outer membrane of the gram-negative bacteria, the outer leaflet has endotoxin. And there are some, in the gram-negative bacteria, the outer membrane has porins and the outer membrane proteins. And the inner leaflet is made up of phospholipids. Then coming to the periplasm, what is periplasm? So you have the cytoplasmic membrane and there is a outer membrane in gram-negative bacteria, and the space between the two is called periplasm, and here it accumulates the components which are exiting the gram-negative cells like beta-lactamases, everything they are located in the periplasm is what you need to remember. Then coming to cell wall of the bacteria. So what is it made up of? Cell wall has a peptidoglycan which provides the rigid support and protects against the osmotic pressure damage. Coming to cytoplasmic membrane, it is composed of biphosphate bilayer, phospholipid bilayer, and it has got penicillin binding proteins in the cytoplasmic membrane in this phospholipid bilayer, and is also the place where oxidative and transport enzymes, PBPs, which are involved in the cell wall synthesis, they're all located in the cytoplasmic membrane. Now, where do you have lipoticoic acid, doctor? Only gram-positive bacteria have. Lipoticoic acid is not there in the gram-negative. And they extend from the membrane to the exterior, and they are the ones which will stimulate the TNF-alpha, interleukin-1 during the inflammatory response of the body against the gram-positive bacterial infection. Then coming to the gram staining, 2 points you need to know. The thick peptidoglycan layer, which is there only in the gram, typically the thick peptidoglycan layer, that is the one which retains the crystal violet dye in the gram-positive organisms. Whereas the thin peptidoglycan layer, it turns red or pink because of picking up the counter stain. So red and pink is gram-negative, gram-positive is violet in color. That is all the funda of gram staining. So in this one mind map, you get everything and ready to face any MCQ on the cell membrane, gram-positive organisms, gram-negative organisms, what is peptidoglycan. All these things you are now ready to face any MCQ. There are certain bacteria that do not stain well with the gram stain. What are they? Treponema, Leptospira, they're too thin to be visualized. Spirochetes are very thin. That's the reason you can't. Mycobacteria, their cell wall has a very high lipid content. That's the reason they do not stain gram. Mycoplasma and ureaplasma, they don't have cell wall only. That's the reason they cannot. Legionella, rickettsia, chlamydia, bartonella, anaplasma, ehrlichia. They also do not stain gram stain. Why? Because they are primarily intracellular. And in fact, the chlamydia lacks the classic peptidoglycan. So peptidoglycan, which is all there in the gram-positive organisms, it is not there in chlamydia because of the absence of the muramic acid. So what bacteria do not stain on gram stain? Well, and what is the reason? You need to be very sure the next mind map. So see, you can finish 150 mind maps in no time. Last moment before exam, you can't run through notes, doctor. So you should remember that the last moment when the exam time is before you, you have to be ready with the ammunition of the notes in your hand. If it is a running notes, running into paragraphs, you cannot. But if this running notes in the form of mind maps, colorful mind maps, cryptic, precise, focused, and made available to you, preparation is fun. So now only go to the PDF link, doctor, below. Click on it. You will go to our Instamozo store. There you download. It is just 400 rupees is the initial offer of this 153 mind maps. Even if you attend a lot of coaching programs, 3 days microbiology crash revision, 2 days microbiology crash revision, ultimately what you are going to get is the gist of these 150 mind maps. I'm so happy to see 13 online classmates. I'm Dr. Murali Bharadwaj here to stay with you, hold your hand, and make you to walk. Earlier, I used to give 1200 pages notes packed in paragraphs, but that was all very packed. So now I found a new method of presentation in the form of summarized, visualized, analyzed mind maps, which will help directly to drive the high-yield facts into your mind. And you are going to revise the entire subject of totally 6,000 mind maps covering 50,000 high-yield facts within a span of 100 hours. 100 to 120 hours में एक राउंड रिवीजन मारना है आप माइंड मैप्स के साथ. Already biochemistry mind maps is uploaded into the e-book store, and also we uploaded the microbiology mind maps. It's called microbiology snapshots, biochemistry snapshots. So, doctor, let's go back and master the technique of how the mind maps will help you. One single mind map, I gave you all the staining techniques, doctor. Staining techniques. जरा कलर चेंज करेंगे, मूड भी चेंज हो जाएगा. Right? Yeah. So staining techniques. Genes are stained. Chlamydia, rickettsia, trypanosomes, Borrelia, Helicobacter, Plasmodium, genes are stained positive. Periodic acid shift stain. It stains the glycogen and mucopolysaccharides. Whipple's disease, Stropharia whipple, which lead to malabsorption, typically periodic acid shift stain. Gill-Nielsen stain, it stains the acid-fast bacteria, Mycobacteria, Nocardia, even protozoan. Please don't forget, many times this came as MCQ. Cryptosporidium oocysts are also acid-fast Gill-Nielsen stain. Rhodamine, rhodamine stain is used for, once more, acid-fast bacteria. Then India ink stain for Cryptococcus neoformans, musicarmine stain for Cryptococcus neoformans capsule, silver stain is positive for Helicobacter pylori, Legionella, Bacterial, Coccidioides, Pneumocystis, Aspergillus, silver stain. रट्टा मारो बट्टी मारो कैसा भी नीट पीजी का टॉपर बनो. Now, fluorescent antibody, syphilis, pneumocystis, giardia, Cryptosporidium, they're all fluorescent antibody positive, is what you need to remember. Khatam, one single mind map will make you to remember, doctor. Today only download and buy the microbiology snapshots, 150 mind maps. So, doctor, abhi aa gaya the most difficult part of the human life. What is that? It is called culture media. Why we don't like microbiology? We don't like microbiology because, oh, mommy, please give me a break. There are so many culture media that I need to master. You need not worry. Now you have microbiology mind maps, snapshots for you to remember. Just download the PDF and do the revision, doctor. Now let us see. Culture media for isolation. मेरे साथ जैसे जैसे आप भगवान की स्मरण करोगे, भजन करोगे, मेरे साथ भजन करो मेरे साथियों. कल्चर मीडिया के बारे में chocolate agar, chocolate agar, chocolate agar. So don't forget chocolate agar, Haemophilus influenzae. Haemophilus influenza produce two things. One is called factor X. So it is this factor X which is the one that lead to hemolysis in the chocolate agar. Thayer Martin for the Neisseria, both the types of Neisseria. Bordet Gengol, Rigan Loi for the Bordetella pertussis. Tellurite agar for the Corynebacterium diphtheriae. Loflus medium Corynebacterium diphtheriae. Eaton sagar, कौन ईट किया? कोई भी ईट नहीं किया. Eaten agar, Mycoplasma pneumoniae. So Mycoplasma pneumoniae, remember, fried egg colony appearance. Now McConkey agar, lactose fermenting enterics. Eosin methylene blue agar for E. coli. Charcoal yeast extract agar buffered with cysteine and iron for Brucella, Francisella, Legionella, Pasteurella. Severoids agar for fungi. So these are the agars. सुबह उठते ही फटाफट फटाफट रिवीजन करना डॉक्टर. मुरली भारद्वाज का माइंड मैप्स का जादू देख रहे हैं, right? So there should be some easy way, doctor. Otherwise, people cannot master 50,000 high fetch you like that. Lot of toppers, if you ask, they have the method of writing their own notes. Last moment, ratta marne ke liye. So, what are the indicators? If you take EMB agar, the E. coli colonies have a green metallic sheen. On the McConkey agar, lactose fermenting enteric organisms produce a pink colony. Then what are the selective growth conditions? If you look at the Thayer-Martin agar selectivity, what is meant by selective? One type of organism will grow, other type will not grow. That's called selective medium. So the gram-positive organisms typically are inhibited because of the vancomycin, whereas the gram-negative, except Neisseria, are inhibited with trimethoprim and colistin, and fungi is inhibited from growing by nystatin. So all gram-positive because of vancomycin don't grow on Thayer-Martin. All the gram-negative organisms, they don't grow because of the trimethoprim and colistin, whereas the fungi will not grow because of the nystatin in the Thayer-Martin. Only Neisseria will grow. Neisseria will grow. That is the reason it is called selective growth, selective medium, right, doc? Now, there are some special growth requirements. Special requirements. What is special? For H. influenza, it produces factor 5, NAD plus, and factor X, which is hematin. Don't forget one of the favorite MCQ of the examiner, doctor. Eaten agar is for mycoplasma. Then how does mycobacteria grow? Lowenstein, Middlebrook, rapid automated broth culture, they all will grow the Mycobacterium tuberculosis. In one mind map में आपको सब कुछ मिल गया, right? सब कुछ मिल गया, doctor, right? So once more, I like to tell you that preparation for neetpg, you 120 hours में 50,000 high yield facts को कैसे revision करोगे? Dr. Murli Bharadwaj, 6,000 mind maps, right? 6,000 mind maps covering 19 subjects. Already biochemistry ka 150, microbiology ka 150. It's called Snapshot series. Biochemistry snapshots, microbiology snapshots. It is all available, मेरे यारों. नीचे आपको वो PDF डाउनलोड करने वाला लिंक है. 400 रुपीज पे करना, आउट स्टोर से डाउनलोड करो और आपके आईपैड में लगाओ और मजे में रिवीजन करो. कोई कोचिंग का भी जरूरत नहीं पड़ेगा आपको. So every day we meet, we study together, and one day we tell the world that we made it to happen in the neetpg exam.